Now, the capsule is also known as the slime layer. It's composed of a layer of proteoglycans, and the proteoglycans is a mixture of proteins and polysaccharides. These proteins and polysaccharides are going to control the exterior environment to the cell, and they will keep that cell moist, and that's why it's called a slime layer. They will thicken the outer layer, and they will also control the environment. So they will maintain moisture content, and they will also prevent and create they will also prevent drying out, creating biofilms. If you've ever seen, quote, a slime mold, that is many bacterial cells together, all of whom have capsules. So a cell wall. Cell walls, by the way, are composed of similar stuff. They're composed of proteins and carbohydrates as well. But instead of being proteoglycan, they are peptidoglycan. So peptide and glycogen. So peptidoglycan, it's still protein and polysaccharide. Now, that is different from the cell wall of a plant cell, which is cellulose, which is all polysaccharide. Other organelles that we have, non-membrane bound organelles, by the way, because a prokaryote will have no membrane bound organelles. That is a key feature here. No membrane bound organelles. Another feature you might notice will be an inclusion body. An inclusion body is anything that is, quote, included in a bacteria. It might be something that bacteria ate. It might be an artifact of the preparation process of the slide. It might be some other piece of bacteria that, it might be some injury to that bacteria from an invader or an attacker. But it is an inclusion body. It is not a membrane-bound organelle. It is not bound by any membrane. It has just been engulfed by the bacteria, or it is part or embedded in the bacteria somehow. Once again, about those ribosomes, there are 70S ribosomes. I think we covered that. We did cover that, yes. All right. Now, other things you might find. Plasmids. Plasmids are circular strips of DNA. They are separate from the nucleoid region, the main DNA circle. And by the way, the nucleoid region is circular, naked DNA. And when I say naked DNA, I mean this DNA is not bound by histone proteins. So they are not bound either by a nuclear membrane or nuclear envelope, nor by histone proteins. They are just squiggling around in the soup, doing their thing. Plasmids are small circular pieces of DNA separate from the main nucleoid region. Flagella and cilia. These are motile extensions of the prokaryote, which are composed primarily of microtubules. And flagella are longer, and they have a whip-like motion, whereas cilia tend to be shorter, and they have a back and forth waving motion. Now, cilia need to be distinguished from fimbriae. Fimbriae right here. Fimbriae are little hairs. These are like the sticky part of Velcro, or, well, if we're going to go with non-branded stuff, hook and loop fabric. So the fimbriae, those are helping them to attach to other structures and walls and stuff. And let's see, what else? Yes, the pilus. The pilus is one of my favorites. The pilus is actually a modified fimbriae. The pilus is actually, bacteria can use this for a number of different things. My favorite being the conjugative pilus. The conjugative pilus allows bacteria to undergo a process called conjugation. Conjugation in the, how shall I say this delicately? Adult sense. Yeah. Conjugation is the bacteria can swap DNA back and forth in order to trade traits back and forth. There is some penetration by the pilus, as it were. So my favorite pilus, by the way, is the one most well studied in E. coli bacteria, called the F pilus. And before you think too hard about that, no, the F does not stand for that. The F stands for fertility, because of course it stands for fertility. How dare you think that, you dirty-minded children. All right. That's woohoo indeed. So here's just a list of very much everything I said previously. Yes, even my little joke right here. All right. So here's cilia, cousin it, and here's flagella, walkers from, you know what? No, I'm not going to say what's shown. All right. So here is cousin it. These are going to move in this hair-like pattern. There will be an ultimate movement. But this allows for bacteria to be mobile. They do not require this. There is no requirement for bacteria to have either cilia or flagella. And it doesn't require you to have one or the other. You can have both. There's no reason why you're limited in such a way if you are a bacteria. However, it is simpler to have just one. Now, that said, how do you move if you are either cilia or flagella? The flagella has a propeller motion, and the cilia has a back and forth whipping motion. They have a back and forth beating motion. There are three overall basic shapes for bacteria, and these are all scanning electron microscope images, by the way. You have cocci, you have bacilli, or rod-shaped, and you have spirilla, or spiral-shaped bacteria. So streptococcus, or meningococcus, anything that ends in coccus will be a cocci bacteria. They are spherical in shape. Anything that ends in bacillus, like lactobacillus that you might find in your yogurt, or whatever it is Jamie Lee Curtis is eating these days. Those are rod-shaped. Also, Bacillus anthracis, anthrax, is a rod-shaped bacteria. It's also a spore former, but that's for another day, or perhaps never, because, you know, anthrax. So bacilli are rod-shaped bacteria. Spirilla are spiral-shaped bacteria. Spiral-shaped bacteria are spirochetes. Now these bacteria, some examples might be Lyme disease. Lyme disease is caused by rickettsial illnesses, and that would be a spiral-shaped bacteria. Also syphilis is caused by Treponema pallidum. Yeah, Treponema pallidum. If I'm wrong, the comment section will surely let me know. So those are spiral-shaped bacteria. Mycobacterium tuberculosis is one that we'll be talking about in Unit 5 considerably. These are rod-shaped bacillus bacteria. Now, this is a scanning electron false color microscope image with a nice blue background, a little bit of yellow right over here. It's very tasteful. This is a stained, acid-fast staining image of light microscope variety. They're stained red against a pink background. And then we have a cartoon drawing here showing the mycolic acid over here. These mycolic acid spirals moving out here. Now that's for another day. Now these are the symptoms of tuberculosis. This is by an artist, Jorge Muniz. I probably am going to need to cut this out because this is actually copyrighted. So yeah, note to whoever edits this, cut this out. Cut this out.
Cut it out.
Okay.
Wait.
Oh wait, so you're sure about this? Okay.
Yeah.
Wait,
Yeah.
so we talk in front of the camera now?
In fact, you know what? Stop the recording so
wait,
that we'll
Thomas,
know
talk
to
in
cut
front of
this
it.
part out.
Okay.
And then restart it.